This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 130, recorded on April 22nd, 2011. Hi, everybody. I'm Vincent Racaniello, and it's time for TWiV, the podcast where we talk about viruses. Joining me today from Western Massachusetts, Alan Dove.
Pleasure to be here.
Good to have you back. How you been?
Oh, doing okay.
Working hard or hardly working?
Working hard lately. Actually, been keeping pretty busy.
Yeah? Well, Are you planting your garden?
Well, no, this is going to be the year that we supposedly get the rest of the landscaping around the house fixed up. So,
all right. Also joining us today from down the coast in north central Florida, Rich Condit.
Hi, fellas.
How you doing?
Hey.
Hi, Vincent.
What's the temperature in centigrade there?
I don't know what it is in centigrade. I'm sorry. Centigrade doesn't make any sense. But, it's interesting because in the laboratory, I think in centigrade, and outside of the laboratory, I think in Fahrenheit. And they don't interconnect.
Yeah, it is. A number of years ago, I converted my external temperatures to centigrade. So in my car, I have a thermometer.
Is that right?
And you can convert it to centigrade. And, in the morning, I drive my kids to school. I say, it's 4 degrees centigrade. And invariably, they're like, what's that in Fahrenheit? I say, figure it out, guys.
I can see them rolling their eyes, saying, get a life, Dad, right?
I like centigrade. I can tell how close to zero you are. It's really good. And that's it for today. No Dixon. I have no idea where he is. He's been gone all week.
AWOL, huh?
He's AWOL. We'll proceed without him. This week has been quiet. It's like a holiday kind of week.
Except for the compressor.
Yeah, I have a compressor above my office that's very noisy today. Something's wrong with it. And I threatened to go and smash it with a bat, but I don't have a bat to stop it from being noisy. So if you hear
Besides,
any noise,
bats are endangered in a lot of places.
That's right.
Would use an aluminum one. But then the cold room would die. But I don't have anything in it, so I don't really care. Rich, is the sun out down there? Just want to know that.
Yes, it's bright blue skies. Few puffy clouds, 85 degrees Fahrenheit. I don't know what that is in centigrade.
Okay. Well, here it's really cloudy. That's why I ask.
It's really nice here.
Not
this
that it's
is a good time of
problematic.
year. Good time of year.
Well, we are set to talk about viruses, and we have a couple of neat stories as usual. In fact, we have this agenda that we use to look at during the show, and we have future stories which just keep adding up. We have like 10 now, and there's just so many cool things going on in virology.
Absolutely.
And the first one happens to be on a virus which I have worked on. We continue to work on related viruses, and these are rhinoviruses. The name of the paper is Culture of... Well, that's not the name. Let me actually go to the paper.
Molecular Modeling, Organ Culture, and Reverse Genetics for a Newly Identified Human Rhinovirus C.
Right, a new set of rhinoviruses. And before we go on, let me just object to the name Reverse Genetics, because I'm an old school guy. I made the first infectious clone of a RNA virus. Okay?
Right. So what's wrong with reverse genetics?
It's just genetics. Rich, you should appreciate that. You make a mutation, you get a virus. That's genetics. Why is it reverse? Just because you start with DNA and go to virus, who's to say it's not forward and what we used to do is reverse?
Yeah, well, it's historical,
It just bugs me that someone named it 10 years after I did. I did the experiment. They decided to do it with another virus and name it reverse genetics. It bugs me.
Okay. So do I have to change do I have to change my chapter now?
You don't have to change anything. I'm just ranting.
Good. Okay.
Yeah, I always just found the term reverse genetics in any context kind of annoying.
I mean, I do think it's just genetics in the end, right?
Yeah, because the whole point is to connect phenotypes and genes and understand them and how they interact. And, and this whole forward versus reverse is just a layer of confusion.
So this paper deals with a new set of rhinoviruses that have only been discovered in recent years. So let's start at the beginning. Rhinoviruses are members of the picornavirus family, which also includes myvirus, poliovirus. These are small icosahedral RNA-containing viruses. And since the 1950s, we have known...
Positive sense RNA
Positive
genome.
sense.
Let's do the whole number here.
Yes, I was on the street the other day and ran into Ian Lipkin, and he said, "What viruses are you working on these days?" I said, "Rhinovirus, EMCV, poliovirus." He said, "Oh, you're staying positive."
Oh, bad.
All right.
So it says here that I'm looking at viral zone. Rhinoviruses have recently been merged into the enterovirus genus.
Oh, that's correct. So the picornavirus family is divided into many genera, and we used to have an enterovirus genus, which included viruses that were spread fecal-orally and initiated infection in the gut, and then there were rhinoviruses because they were respiratory and a number of others. Recently, with all the sequence analysis, they've all been reclassified because it turns out that the sequence allows you to classify them differently.
Mm-hmm.
So now the polioviruses, all the enteroviruses and the rhinoviruses are now in one genus called enterovirus, which is a problem for teaching because the name doesn't make sense for the rhinos. But,
right.
I mean, they should give it a different name, I would think. And so, but they haven't. So now we have, because it turns out that polio and rhinoviruses genetically are quite highly related, so they should be together.
Yeah, but this gets back to something we discussed many episodes ago about taxonomy and
right.
The molecular versus ecological taxonomy, and it's not irrelevant what disease they cause. I mean, just because they have a similar sequence, it's conceptually useful in some ways to separate them.
Yeah, I mean, they were subjected to different selective pressures at one time.
Yeah,
so the rhinoviruses were a separate genus, and the enteroviruses were a separate genus,
right?
And that was based on disease profile, really, right?
Yeah, disease, tropism, antigenicity. Okay. Yeah,
the rhinoviruses cause respiratory disease. The enteroviruses cause gastrointestinal infections.
Yeah, some of them cause respiratory disease also.
Okay.
So the rhinos were identified in the 1950s, and we know they cause common colds, which they have that name because they're very common. They happen very often, and rhinoviruses account for about half of all common colds.
And that's why they're called rhinoviruses.
Yeah, yes, that's a good point. They were originally believed...
These are not viruses that affect rhinos, although rhinos probably have similar viruses.
That's right. They were originally believed to mainly infect the upper respiratory tract, so they were called rhinoviruses. Now, the original rhinos were isolated in the 50s by taking nasal secretions from people with colds and putting them in cell culture. And by doing that, they grew out slightly over 100 different rhinoviruses. So we used to say there were 105 serotypes of rhinovirus. Now, a number of years ago, not too long ago, I would say 3-4 years ago, 5 years ago, people started looking in individuals with severe lower tract respiratory disease and just sequencing samples from the lungs. And they pulled out a number of new rhinoviruses which had not been discovered before because they weren't able to be cultured. So from the 50s and 60s to the present, we didn't discover these new rhinoviruses because you couldn't grow them in cells. So we had sequences of them. And now when you take all these sequences and compare them now, everything's been rearranged. We now have 3 what are called species of rhinovirus, A, B, and C, Rhino A, Rhino B, and Rhino C. And A and B, Rhino A and B are basically the rhinos that were around that we knew about since the 50s and 60s. And the Rhino Cs are the new ones which can't be grown in culture and which seem to be associated with very severe lower respiratory tract disease.
So you also sent along to us this older paper from the Lipkin lab that does a lot of phylogenetic analysis on these things. Mm-hmm. And they have a tree in this that's HRV-A, HRV-B, HEV, and a novel clade. Is the novel clade this HRV-C?
Yes, this is HRV-C.
Okay.
So this is an older paper, right?
All right. So there's a bunch of them. There's a bunch of them.
Yeah, there are a bunch of them. And there's many, I think... how many do we have? 48 different HRVCs now.
Okay.
Was the latest.
This is why you can keep getting the common cold every year.
Yeah, so we're up to like 150, 200 different
types,
viruses,
and now we call them types. So we can't say serotype anymore because these new ones can't be grown. And as I've been told by the people who work on these, the old serotypes were defined by putting virus into a rabbit, getting the sera, and see what it neutralizes. And it's not relevant to what happens in people. And so we don't talk about serotype, we talk about types or genotypes.
Nevertheless, the idea is if you get a cold caused by one type,
yes,
you are not necessarily cross-protected against infection by the other type.
Exactly right. And there's a certain percentage of sequence difference that defines a type, and that's assumed to be, won't lead to cross-reactive antibodies, although those studies will have to be done with the C viruses.
So right, and I think it's worth pointing out in medical terms, people think, oh, the common cold. Well, actually, who really cares because it's not that serious a disease. But this, in terms of the societal cost, it's actually it's way up there.
Yeah, and also it's a very, very significant inducer of asthma.
Yes.
So because rhinoviruses cause so many infections. They cause a lot of asthma exacerbations. 50 to 85% of asthma exacerbations are due to rhinovirus infections. Wheezing illnesses in infancy are associated with a high risk of developing asthma. So they're pretty important, and people have been working on them to try and understand how all this works. So the problem with the rhino Cs is that no one has been able to propagate them in cells, and that's hard to study. They become hard to study when you can't grow them. You can't titrate them. You can't do plaque assays and all that stuff. So in this paper, they achieved that to a certain extent. And they've tried many, many different cell lines of different sorts of human cell lines, of respiratory tissues and so forth.
Right.
They have taken nasal specimens
from
patients and put them in various cell lines, and the virus doesn't grow.
Right.
So what they have done here, which seemed to be important, is they tried growing these viruses in surgery-derived byproduct tissues from sinuses, adenoids, tonsils, nasal polyps. So people have various surgeries done on their upper tract, and you get little pieces of tissue, and you can get those, and otherwise they'd be thrown out. And so they got a bunch of these and tried it. And who found this interesting diagram?
That was me. So, because, I'm always interested in the methods, and they describe in the methods how they got the sinus tissue. All protocols approved by the Health Sciences Institutional Review Board. We obtained residual airway tissue specimens from individuals with chronic sinusitis undergoing endoscopic surgery. Informed consent was not required, as specimens would otherwise have been discarded after surgery. And so I was interested in kind of what the circumstances are there. And, there's a lot of people with chronic sinusitis involves, among other things, extra bits of tissue growing here and there in your sinuses that cause obstructions. And so they have endoscopes that they stick up your nose along with other instruments to snip away this tissue and extract it to make your breathing easier. And apparently this goes on all the time. And so they can recover this tissue. So I wanted to know what this looked like. So I, Googled chronic sinusitis surgery and looked at the images, okay? And you get an amazing assortment of images. And even though the one that I put here is not entirely accurate, it gave me an idea what was going on.
Yeah.
So they got these bits of tissues, and then they infected them with nasal washes from patients with Rhino C infections.
I want to go on here because organ culture interests me. They take these chunks of tissue and stick them in dishes and just cover them with growth media. So it's basically just a chunk of tissue floating in a plate in the regular cell culture growth media. And you can keep the tissue alive, in quotes, for a period of time doing that. So that's what they call organ culture.
And
it's actually in the pictures. It's kind of cool. Instead of seeing a cell culture plate, you see this just little lump of flesh essentially floating in the dish, and then they infect it and get the virus in.
You know, it's interesting. Before we had cell culture, people used to do this to try and grow viruses. And it's really hard because the viruses often don't spread throughout the clumps, and the clumps don't multiply; they die. But now we're going back because this is the way the cells are in the tissue, right? Right,
and there are viruses that require the three-dimensional architecture of the tissue in order to undergo a complete replicative cycle. They need the cell-cell interactions and et cetera. And when you make cell culture, you disaggregate those. We just talked about one of those recently when we talked about papillomaviruses. Also, I think this is some of the beginnings of cell culture in this, because lots of times if you start an organ culture like this and keep it around for a while, the cells will grow out of the organ onto the dish.
Right.
Okay.
Yeah, that would be good. They could make a cell line from it, and it might be susceptible.
Right.
So if you infect these bits of tissue with these nasal washes, you get viral replication. Now, how do you assay viral replication? You don't have a plaque assay, so they have to measure the viral RNA by PCR. All right, so then they talk about viral RNA copies per milliliter. And the virus seems to replicate in these organ cultures. They can get production of virus with time. They can take the yield and pass it to another organ culture, and it will infect that. So it looks pretty good. And the different bits of tissues have different efficiencies, probably. There's a lot of heterogeneity also from different patients, I would assume. But it seems to be replicating. I'm convinced by this. Mm-hmm. And they also have some nice staining where they stain for viral RNA in these tissues, and you can see they stain it purple. You can see the cells that the virus is replicating in. What I found cool is they said during each passage we observed partial inhibition of ciliary beating, coinciding with the peak of virus replication. Right. So these organ cultures, they're really differentiated. They have ciliated mucosal epithelia, and the cilia are beating.
Right.
Of course, that's the normal function in your respiratory tract, to move mucus out and move the junk out that they've filtered out.
Yeah, this tissue still thinks it's a sinus.
It does. And this virus likes it, apparently.
And it's not uncommon for respiratory viruses to trash the ciliated epithelium to the point where the cilia no longer work, and that's responsible for some of your symptoms.
Right.
You can't clear the secretions anymore.
Right. So to me, this suggests, and to them as well, that the cells in this organ explant have a receptor, most likely, or something else that the virus requires. And that's why it hasn't replicated in the other cell types. So they do a very extensive sequence comparison. For the Rhino A and B, we know they bind two different kinds of receptors, ICAM1 or low-density lipoprotein receptor, and we know, and those have been structurally solved together with the virus, so they know where the receptor sits down on top of the viral capsid. And they look at the sequences of these Rhino Cs, and they see that they're missing a lot of residues that would normally be interacting with the receptor if they were a Rhino A or a B. So they say, it must be a different receptor. And I think that's a reasonable hypothesis. So that is quite a bit of the analysis in the paper. So their conclusion, overall, the analysis predicts the Rhino Cs use a different receptor with a distinct compositional footprint from Rhino A and B. And
so that'll be the next project.
That's right. In fact, they're working on it. And
that'll be tough, though, with no culture, with no cultured cell system.
Well, this is right up your alley, right, Vincent?
Yeah. Well, I was emailing Ann Palmenberg this morning, who's one of the authors on this paper, and she said, from the structure, they got some clues about what the receptor might be, and they're chasing those down. They think they're on the right path. So, cool. How do you do with that? Well, you put it into cells, and you see if you can get multi-cycle virus replication. Now, how do you do that? Okay, so that's the next part. That's their, quote, reverse genetics, unquote. So they take a full-length DNA copy of the genome, they put it in a plasmid with a promoter, and this would be similar to the T7 RNA polymerase promoter that Rich Condit reminisced about.
Yeah, I think, in fact, yes, T7 promoter that they used.
And they use that to make RNA in vitro, and then they take the RNA and they transfect it into HeLa cells and Wis-L cells, and you can see you get cytopathic effect. You get virus production, again measured by PCR. So it's one cycle of infection, right? Because the virus that's produced can't reinfect these cells because they don't have the right receptor. And they actually do an EM on the yield, and there's viral particles in there. So the clone is infectious, which is good. So now you can take these viruses that are produced and put them in cells where you've expressed various proteins that you think might be the receptor. So I think that's going to be pretty straightforward. You could actually do a whole genome screen, put a whole genome library into cells and ask which protein is a receptor.
That's true.
So we're going to see that within a year, I think. And that'll be cool because then they can put the receptor into HeLa cells, and then you can do virology with these viruses.
How difficult is that going to be if you can't grow up reasonable amounts of the virus to start with? They're taking the virus that they have is from nasal washes from people who were infected, right?
Well, no, they have this clone now.
I guess they have the clone so that they can make some virus, yeah.
So they say they can get 10 to the 9th copies of RNA per milliliter. So I don't know how that relates to infectivity, but you could calibrate it with another rhinovirus that does replicate and get an idea of how much that is. So I think you could make enough that way so that you could do a screen. But they said they have they've been guessing at what it might be, and that often works as well. So we'll have a viral system that can be used to study this, because organ culture you can't use organ culture to do virology.
No.
Right? It's just not homogeneous. It's too hard to get these bits. Not everyone can get sinus bits, unless you're at a medical center. So they have to make a culture system, and I think they're on the way. So this is pretty exciting because these viruses are important. They cause serious disease, more serious than A and B. So we need to study them.
Yeah, it's not something to turn your nose up at.
And as
Thanks, Alan.
I can tell you that as soon as a virus cell system is available, I will get it, and we will work on it. A lot to do. So there you go.
All right.
A genetic system for rhinovirus C.
Good.
Cool.
Let's see what we have up next here. The next paper is right up Rich Condit's alley. Actually, we have two of them. Vaccinia transmission. One is among wrestlers and another in a martial arts gym. And we have talked about some kinds of transmission before, right?
Right.
I think it was sexual transmission.
I think we've done a couple of these before. One was a woman who was intimate with a military guy and she wound up with lesions on her vulva.
Yeah.
And then there was another case, I don't know if we did this or not, of a soldier who was vaccinated and came home and inadvertently infected his wife and his son. And his son was an infant who had eczema, and that turned into a really, really nasty case. And then there was another one not long ago of a guy who was, and I forget whether we did this one or not, a guy who was vaccinated, and it turned out subsequently, they didn't know this when they vaccinated him, that he had leukemia, which left him immunocompromised, and so he got sort of a disseminated infection.
Yeah.
So we've had some of these, and some of them being this contact transmission. So,
you want to go ahead and do this,
sure.
Rich?
So this is an article from Emerging Infectious Diseases. I think this is the same volume that came in my mailbox 2 weeks ago. That's got Jenner on the cover, and it describes the title is Secondary and Tertiary Transmission of Vaccinia Virus from a U.S. military service member. So let's remind everybody about, very briefly, about vaccinia and the vaccination, because that's relevant to this. So vaccinia is the virus that was used to eradicate smallpox. Large, complex, double-stranded DNA-containing virus replicates in the cell cytoplasm. The vaccination is unusual as vaccinations go, because what is done is to introduce some of this virus into a person's skin by putting a solution of virus onto the skin and then poking it with this special needle to just barely break the skin, and the virus gets into the skin and starts to replicate, and you get an immune response, and the result is a blistering lesion that's very characteristic. As a matter of fact, a word that's used repeatedly in these articles is umbilicated. So when the thing gets to be about, half a centimeter in diameter, there we go, I'm in metric there, half a centimeter in diameter, there's a little indentation in the middle of it, which is why they describe it as umbilicated, and that's very characteristic. And it's a pustule, so it's filled with pus, and inside there is live virus. So this is different than, your common vaccine where you're injected with something. Even most other live vaccines some of them spread, right? Because you shed the polio virus vaccine.
Sure.
So you can spread that. But in this particular case, what's supposed to happen is when you're vaccinated, you are cautioned by the physician that the vaccination contains live virus, and that can be transmitted to another person, because it will, the virus will infect any break in the skin or any mucous membrane, your eye, your vagina, your mouth, other mucous membranes. And so when you change the Band-Aid, you're supposed to be careful about throwing the Band-Aid away. You're supposed to keep it covered. You're supposed to wash your hands after taking care of it. There's a bunch of precautions that you are alerted to. But apparently, some people don't pay a whole lot of attention to that. And in this particular case, this is in February of 2010. Oh, the other thing is that the military still give people smallpox vaccinations. So smallpox has been declared eradicated since roughly 1978, 1980. Vaccination has been not only not required, but discouraged worldwide, because the risks of problem with the vaccination are greater than the risks of getting smallpox. But the military still vaccinates people, and you can draw your own conclusions from that, routinely. So this is in February. A military service person who had a fresh vaccination, and let's see how fresh here. They've got a timeline of what happened. He was vaccinated on February 23rd, and then he's apparently an amateur wrestler. So he went wrestling on the 27th. Now at that point, you're just starting to grow a pustule. So he's got live virus on his arm. And they say that apparently the covering fell off. That's no surprise when you're wrestling. And he managed to wrestle with a couple of people, and in the process transmit it to them. And they got some pictures here. One guy wound up with lesions all over his face and his neck.
It's nasty looking.
Yeah. And another guy on his arm or trunk.
It's interesting, though, the guy with them on his face, in one of the pictures you can see he's smiling.
Yeah, he's probably pretty pleased. It's nasty looking, and they itch. But in particular, once you know what it is, they say, well, it's just going to heal up. Don't worry about it. Though it may leave some scarring.
And by the way, you've been immunized against smallpox.
That's right. Well, that was the intent to start with anyway.
If you have immune problems, you could have a very serious infection, right?
Absolutely. It could be a problem.
I think one of these people, they gave varicella immune globulin from the national stockpile.
Vaccinia immune globulin.
What did I say?
You said varicella. That's okay.
That's what's written here,
is that what's written? Oh, my goodness.
Yeah.
Huh.
From the strategic national stockpile.
So varicella, I wonder if that's a misprint or if that's what it's actually called. Because I usually think of it as vaccinia immune globulin.
They say it twice. They rate varicella twice, and they have the reference here, number 5. Let's see. Medical management of smallpox, vaccinia immune globulin. It's a misprint.
Yeah.
Okay.
Yeah.
All right. Good.
So there's a national stockpile of that in case of an outbreak.
Actually, and so what that is, it's immune globulin. harvested from individuals who have been recently vaccinated.
Right.
Okay, so they should have a lot of antibody to this virus. So it's a passive immunization. You're injecting them with this antibody. And we're talking about a lot. Okay, it's not fun. And
I assume it's screened for other viruses, right?
I would assume so.
Yeah.
I don't know how effective it is either.
Yeah.
It
worked for this patient.
Well, the patient healed. This is anecdotal.
Yes,
right?
Exactly.
they got the immune globulin and they healed. So at any rate, there were transmission to a total of 4 patients, and 3 of them I think were direct contact through the wrestling, and the other was a woman who was a roommate of this person who got it on her face, and the assumption is that what happened was they shared a face towel. And so he dried his face, she got virus on the towel, she dries her face, she gets it on her face. Okay, so that's a classic example of what we call a fomite, an inanimate object that can transmit an infection from one individual to another.
So when it went into her nose, that's when they got scared and they gave her globulin, right? So that could be it could go into her lungs, I presume, right?
Actually, the interesting thing is that there are no published examples that I'm aware of respiratory infections with vaccinia, though it's not out of the question at all.
Okay.
So that could theoretically be a problem. There are other things that they can do. There are a couple of experimental drugs. There's cidofovir, a nucleoside analog, that has been shown to be effective against poxvirus infections. And then there's this other drug awaiting approval from the FDA. That may never happen, but you can get it on an emergency basis, and that's ST246. And we've seen that used in a couple of cases. One of the things that really interests me about this is that in almost every case, the primary care physicians missed it. They saw people with lesions, and they did not diagnose it correctly.
Well, you don't look for zebras, though.
Exactly. They sent them to dermatologists. The dermatologist got it right and immediately notified the appropriate people.
I thought it was
a lot more wrestlers are going to show up with a staph infection, and a lot more members of the general public are going to show up with either that or herpes infection, which would be for primary care physician pretty easy to confuse with this.
Right.
And the rate is pretty low, 5 cases per 100,000, immunized people.
Right.
That's low.
Right. So actually, yeah, your normal primary care physician is never going to see this.
Yeah, that's why it's. This one little outbreak is publication worthy.
So there you go.
But why did the guy wrestle? I just want to know.
I don't know.
He probably didn't. No, not in that sense. But
why?
After being immunized, not in general. Why did he not heed the advice of whoever immunized him?
Well,
he probably said don't. He probably didn't tell him don't wrestle. But if he's saying don't get in contact with people with your lesion, he should have thought, that includes wrestling.
I would think so. Either that or he, in fact, was, although I think they say that he was counseled appropriately when he was vaccinated. And they probably get a sheet of paper and stuff. I guess he chose to ignore it.
Well, that happens a lot when you counsel individuals either for immunization or if they're in a clinical trial for a HIV vaccine, you have to counsel them not to have risky sex.
Right.
Yet the only way you find out that whether your vaccine is effective is that they do have that. So people always ignore what they're told, I suppose.
Yeah. So this really it underscores 2 things, which is the importance of that advice, but the other thing is the importance of the ongoing work on alternate vaccines for smallpox. Because if we are going to continue vaccinating some portion of the population, then it'd be nice to have a better, more modern vaccine to do it with, probably.
Right, and the 3rd generation vaccine, the MVA, fills that bill.
And it's more attenuated, so it's not going to do this, right?
Right. And it's not only that, but it's administered by injection rather than by scarification. So, I mean, it doesn't even go through a complete replication cycle in humans.
But it is protective, right?
But it is protective.
Okay.
So, and I don't know whether that's going to be used routinely in these sorts of cases or not. It's being developed primarily so that it can be used in people who are contraindicated for vaccination. That is, people who are immunocompromised, AIDS patients, etc.
Yeah.
Should it ever be necessary.
Okay. All right. Tell us about this second one.
So this second one that you just sprung on me,
sorry,
at the last minute, right
before
the show. It's okay.
I figured you could handle it.
I should be on top of this. So this is similar, I guess. This paper is also in Emerging Infectious Diseases, same volume.
Yes.
Yes. And it describes several.
Four.
Yeah. 4. I thought there were even more.
Cluster of 4 cases. Yeah.
4 cases of vaccinia infection associated with a martial arts gym in Maryland. So it's a different locale and et cetera. And basically, these are 4 different people who all study martial arts at this one gymnasium, all of whom came up with vaccinia infections, similar kind of deal. Now, did any of these I don't think any of these had direct contact with each other.
No, and neither were they military, so none of them were immunized.
So the bottom line is they never figured out where this came from.
Yeah.
Okay, they did. There's an interesting side note here. One of the people, I think it was case patient 1, that when they interviewed him, they said, were you ever vaccinated? You've been in the military, whatever. And no. But I was in Brazil recently.
Yeah.
We've talked about before, in Brazil, they have had issues with a cowpox-like disease that turns out to be vaccinia that is circulating in the wild in cattle and transmitted to humans who milk the cows. But that particular vaccinia has a very characteristic genetic signature, and they actually looked at this virus coming from these people and sequenced the hemagglutinin gene, which is diagnostic for these things, and determined that the virus that these people had was this new vaccine, ACAM 2000, and not the stuff that's circulating in Brazil. So, and they never figured out where it came from. So somebody must have, at some point, come to this gym with some lesions and left some virus behind.
Yep.
And several other people studying martial arts in the same gym picked it up, and they don't know how or what it came from. Very interesting. Same sort of drill, where they go to the primary physician. The primary physician sends them to a dermatologist. Dermatologist says, this looks like vaccinia, and alerts the appropriate public health officials, and they do the appropriate diagnosis, which is sequence determination, and figure it out.
Yeah, it's an interesting counterpoint because they didn't figure it out.
So the one interesting thing here is that we were talking about earlier is this thing. They say the CDC identified 5 civilian clinics that had received ACAM 2000 vaccine, that's this vaccine, since late February in the Maryland area. And these clinics reported having vaccinated 65 persons. None were members of the martial arts gym. And also the military vaccine agency cross-checked its list of vaccinees, and none of them went to the gym. So I was interested that 65 people had been vaccinated in the Maryland area since February. As Alan points out, there's laboratories in that area that work on vaccinia, so maybe that explains it.
Well, and there's also, the area is chock full of military contractors and quasi-military agencies. I mean, the NSA is there. I mean, maybe I'm not supposed to tell you that, but a whole bunch of, I think the National Reconnaissance Office or whatever it is that does all the satellite spying, they're based in Maryland. So there are a lot of organizations that would have civilian employees or at least non-military employees who might need this vaccine for reasons they can't discuss.
Interesting.
So anybody out there who has any occasion to be in touch with somebody who gets a smallpox vaccination or who gets a smallpox vaccination themselves, be aware that those precautions that they tell you about are not a joke.
Yep.
The lesion has live virus in it, and if you're not careful, you can transfer it to somebody. And usually it is of no great consequence, but it's at least annoying, and it could be, if you transfer it to somebody who's got eczema or is immune compromised, or if somebody gets it in their eye, it could be a serious problem down the road.
Yep.
Very good. Okay, let us move on to our final story, which is a new era for treating hepatitis C virus. 2 papers came out in the New England Journal of Medicine, phase 3 clinical trial of a new antiviral compound. Hep C, of course, we have talked about on TWiV, discovered in 1989. 170 million people infected worldwide.
Let's do the virus, too. So, right.
RNA positive strand, right?
Right.
Flavivirus, enveloped.
Right. So it's got an icosahedral capsid and it's got an envelope around it. So, and it has the genetic structure is similar to the coronaviruses. Is that right? How would you evaluate that?
Yeah, it makes a polyprotein, one long protein, which is processed by both viral and cellular proteases.
And the organization is more or less the same, right? With the capsid proteins first and then the replication proteins second?
Yeah, that's correct. So this is spread initially by sex and drugs, basically. Sex and intravenous drug use, and also originally by the blood supply, by transfusions. Of course, it was tested for after the virus was cloned in 89, and then so now there's no transmission by the blood supply, and but there are a lot of people still infected, and they can die. They can get hepatocellular carcinoma. So there's been a lot of effort to make antivirals.
Right. It causes chronic infections. So,
chronic infection, long term, your whole life.
Not all the time.
Not all the time, right? A certain fraction of the time. Some people resolve and others go on to chronic infection.
And if you resolve, you're okay. If you get a chronic infection, you're in trouble because that can lead to hepatocellular carcinoma, liver cancer. And it's the major reason for liver transplants in the U.S. nowadays. It's a big problem.
Yeah, it's, similar story to Hep B in that sense.
Right.
Of course, with Hep B, we have a wonderful vaccine.
We now have an effective vaccine, yes.
So we don't need to have antivirals, although there are some that are effective. So the treatment so far for Hep C has been interferon plus ribavirin. And my understanding is that this is a miserable regimen. It's got side effects and it only works about half the time.
Yep.
That's the number.
Yeah, the interferon actually has some major psychiatric consequences because it's apparently it's incredibly depressive.
Yeah.
Yeah, as a matter of fact, in one of these, this is sort of skipping ahead, but in one of these papers they say there were 6 deaths during one of these trials, and 2 of them were suicides that they attribute potentially to the interferon.
Yep. Wow. So there's clearly a need, and I found a website which is amazing because it lists all the drugs in the pipeline. It's called hcvdrugs.com. And right now there are 6 combination therapies going on and 93 individual drug development projects.
Many of which are phase 2, phase 3.
Yeah, they have a nice graph format where you can see what phase they're in.
Yeah, it's not just a whole lot of basic science going on. It's actually, this stuff is really pushing toward the clinic.
It's amazing. Now, there is the discovery of this virus has spurned all of this clinical research, as well as a lot of basic research. Many labs have grown up just doing Hep C. Many well-funded labs. And now it's interesting, there was a commentary in Science, and it says basically, since the virus was discovered, screening has made transmission rare through blood, and other modes, such as needle sharing, and sexual practices. Infection rates in Western countries have dropped sharply. As more patients are treated or die, the market in rich countries will start shriveling. The companies are all racing against each other and against time, someone from Janssen says. So all these companies, these 93 and more drugs, they have to get their products out. Otherwise, they're going to lose. Anyway, check that out. That's pretty cool.
It is very cool.
I wanted to talk about the discovery of this drug. This is really a really cool story. The name of this drug, we haven't said it, is boceprevir. That's just the trade name that they've given it, but it has a very, very long chemical name. And there's a paper called Discovery of this compound, and it documents. This is open access paper, I think, so we'll put a link. And they say here, so this is an inhibitor of the viral protease, which is NS3, and they had gotten the crystal structure of the protease, and they say that the crystal structure of the enzyme reveals a featureless, shallow, highly solvent-exposed active site. And so they took that and tried to screen for inhibitors, and they didn't get anything. They screened chemical libraries, found no inhibitors. Michael Gale has told me, because this active site is so shallow, it's really hard to find things that will fit into it. There's no pocket for drugs to fit in. So what they did was structure-aided design. So they said, we envisioned trapping the catalytic serine with conventional electrophiles, so that they designed molecules that they thought would go in there. And they got some hits initially from that design. They had one initial molecule that gave a really good inhibition, and then they started to modify it. And this is really cool. Listen to this. Inhibitor 1, though an excellent inhibitor of the viral protease, lacked selectivity against human proteases. Okay? Moreover, with such a large number of peptide bonds in the molecule, it was bound to have poor pharmacokinetics. So they had to modify it to get around these issues. So this is just perfect example of what you need to do to make a good drug.
Well, and this whole rational drug design idea has been around for at least 20 years. And for a long time, the standard response to it was, well, yeah, but it hasn't accomplished anything. And that was true for a very long time.
Yeah.
People were talking about, oh, well, if you have the 3D structure, you can target it. And then people tried to do that, and it didn't work, and it didn't work. And now, we're finally starting to get to a point where this is a viable method of drug design. It's certainly not going to work in all cases, but this is a great example of a case where you can do, or they were able to do this kind of rational approach and get a compound that now appears to work.
Yeah. So this is called structure-aided design, and it was also done for the flu neuraminidase inhibitors, the Tamiflu and Relenza.
So have we said what this protease is for in hepatitis C?
I have not.
Okay. So the way that this genome is expressed is like polio. It's got a bunch of genes on a single-stranded RNA, and there's a single translation initiation site at the 5' end, the start side of the molecule, and you make one big, long, what they call polyprotein. That is all of these gene products made as, well, one big polyprotein. And then one of those products is a protease that can then cleave the polyprotein into all of the individual proteins. And it's that protease that this drug targets. So if you can specifically target that protease, then you'll inhibit the formation of all those proteins and basically stop the replication of the virus. But of course, cells are full of proteases that do all kinds of stuff. So the drug has to be very specific for the viral protease and not inhibit the others, otherwise it'll be toxic.
Right. Right, so the protease is essential, and so if you inhibit it, you can inhibit replication. So this drug was developed, it was shown to inhibit replication, went through phase 1, just safety testing, phase 2, and now these are 2 big phase 3 trials. So one is for people with hep C who have never been treated. They have chronic hep C. And then another trial, another paper, was for people with chronic hep C that had been previously treated with PEG interferon ribavirin. And that had clearly not resolved their infections. and they're very big. They're very big studies. One of them, the untreated one, 938 non-black and 159 black patients. So the blacks were separated because they have a different response to PEG interferon ribavirin.
Right.
And I was looking at the paper that deals with this. This is a really interesting story, which I think we should do separately. Why this polymorphism affects their response. So we'll just say now there is a different genetic difference in that population, which doesn't make them respond well to PEG interferon. So they did 2 separate groups, and they had 3 kinds of regimens. One where you just get PEG interferon the whole time, and then you assess the amount of viral RNA present. And the second group, you get PEG interferon, then you get your drug with PEG interferon ribavirin, so it's always a combination. And then the third group, same thing, except you get treated for longer with the antiviral.
Right.
That's right.
Right.
Pretty complicated. So they do this lead-in period with PEG interferon ribavirin, which is very unusual and made it complicated. So the 4-week lead-in of PEG interferon used in the trials is a major point of divergence from other studies and appears to have certain advantages, but also introduces complexity. So I guess they all wanted to get them to the same point or a similar point by treating with this, right?
Yeah, and part of the deal is they say that there's advantages to doing that lead-in because you get the viral load down with that, and that they figure might not only help with the drug but lower the probability that you'll get drug resistance. I would imagine there are issues here as well. I'm not sure, but I would imagine that there might be issues as well with offering a treatment that works, right?
Yeah.
Yeah.
Yeah, you have to meet the standard of care, too.
Right.
That's called standard of care, yes. So we know these drugs work, so you have to give it to people.
Right, but what's different here is doing the initial lead-in with those drugs and then adding your experimental drug. I mean, to meet the standard of care, you could just start all the drugs at once.
That's right. Mm-hmm. But here they wanted to see, as you said, Rich, whether you could get a better sustained response, but they also say you don't get resistance as readily, and so that was why they did that. The drug works really well. The numbers are good. It really improves. Giving the drug gives you a good viral response.
They assess these patients by basically looking at RNA copies in their blood, right?
Right.
And they have certain criteria for certain cutoffs for whether or not the drug is successful based on how much it knocks back the virus in the blood, and based on that, they got a good response.
So here we go with the SPRINT. Is the serine protease inhibitor therapy 2?
It's the one you were talking about.
Untreated. SPRINT is untreated.
Right.
Here are the numbers. Control group, 38% virologic response. So 38% of the individuals had a good virology reduction in HCV. 66% with 48 weeks of triple therapy and 63% response-guided therapy. So there's not much difference between the 2 different antiviral treatments. So that's a good improvement. So that's SPRINT. And then RESPOND, what are the numbers there? Patients, 7% control, 40% response-guided, and 52% in fixed duration group. The most important finding from RESPOND are the impressive increase in the rates of a sustained virologic response both in patients who had a non-response prior to prior therapy, that's the 7, 40, 52, and in patients who had a relapse, 29% control, 69, and 75 for the 2 drug regimens. So it's really impressive.
Right.
And it leads these, there's a commentary in the New England Journal, they say this is a new era of successful therapy. However, we should point out that if you use this drug alone, boceprevir, you do get resistance. That's why they have to use it in combination therapy. Right?
Even in these trials,
they even do get it here.
People were in the combination therapy. People were throwing off resistance.
Right.
And of course, although it's a step in the right direction, it's not like all of these people are cured. All right. So it seems to me that this is going to work out. And it's similar to HIV, right? We're talking about an RNA virus that mutates very rapidly. And so drug resistance is something that can happen pretty rapidly. I'll bet that in the end, we're going to have a thing like HIV where you've got a cocktail of several drugs.
Yep.
And that's relevant then to this drug pipeline that you're talking about, because it's not like one drug company is going to win the whole show here.
Right.
It's probably, in the case of the HIV drugs, how many are there now? Dozens.
In the pipeline. Yeah, 2 are very close. So this one, and there's another protease inhibitor, TWiV, which are close to being
But they're also talking about polymerase inhibitors and other things, and we're probably going to need a couple of different drugs that hit a couple of different targets in order to get an effective treatment.
Yeah,
and we're going to need alternatives, because even with the multiple treatment, you're going to wind up with multiple drug resistance coming up, and then you're going to need another set of drugs.
And even with all that, there will still be a market for a vaccine.
Yes, absolutely. The vaccine is really what ought to happen ultimately if you can do it.
So we should point out these drugs have side effects, or this one, boceprevir, when we're talking about anemia.
Right.
And some of the people dropped out as a reason. Oh, they had to be given erythropoietin to control the anemia. And so they say, and that's expensive as well. We should point out this is a really expensive drug. I think, what is the price per dose? $3,000 per patient in places where they've negotiated the price down. So the erythropoietin is also expensive. And they say in this commentary, without erythropoietin, additional reduction in the dose of boceprevir or ribavirin would be necessary to manage anemia, which might reduce the rate of sustained response. So
or increase the rate of resistance.
Exactly, yeah. I wonder why it causes anemia. You think it has an off-target effect?
I have no idea.
Well, that's a possibility, right?
Sure.
Yeah.
Or it could just damage red blood cells, I suppose. I think that's a common effect of many drugs. Anyway.
Yeah, anemia is one of those, it's like one of the check engine lights of the body. It kind of happens for a lot of things, I think. So
this goes to show that you have to make a trade-off, right? So this is like a really good candidate. It's effective, but has side effects. So what do? You go with it, or do you go back and find something that doesn't have any? There's nothing without side effects.
No.
Right?
No.
So there you go. So this is pretty exciting. It is a new era.
Yeah, and neat stuff.
And as Rich says, there'll be a lot more. And there'll be combination therapies. And eventually this will be, Charlie Rice says, this is a disease we should be able to lick. And Charlie, I guess that means he'll be out of business one day, right? There'll be no more. So I talked to someone this week who said that SARS funding is now almost zero in the U.S. There's like two grants to study SARS. I don't know if that's true or not, but someone told me that. So can you imagine? Right now, the Hep C grants are big. There are a lot of them, but you can imagine someday they're not going to be a lot either if the disease is controlled.
Yeah, but that's kind of the goal in a lot of medical research, isn't it?
Yeah, it should
put yourself out of business, right?
Yeah, ultimately you want to put yourself out of business, and it's certainly not a problem for the individual researchers because you can move on to other projects then.
So I should be out of business with polio, right?
Sure, but you're working on rhinoviruses and other enteroviruses. There's no shortage of things to work on.
Right.
They told you were going to be out of business some time ago, right?
No, I still work on polio. I don't have a polio grant, but we still do experiments on it. It's hard for me to let go of things.
I don't blame you at all.
Probably going to be my decline, but it's all right. All right, let's do some email. We have a lot left over from last time.
Oh, yeah.
We have one from Suzanne. Of course, in the meantime, many more have come in. All right. Suzanne writes, I know it has nothing to do with viruses and would be a better question for Futures in Biotech if he did questions, but I figure you guys would at least have some idea and know who to ask if you needed to. I've been curious for a while about the techniques used in the creation of genetically engineered crops. Is it the same as bioengineers in medicine? I know these days scientists can do some amazingly precise manipulation of DNA. I also remember hearing that the companies on the agricultural side of it were using older, more random ways of getting new genes into seeds and then growing them to see what works. If so, would this make a difference in the outcome? Would it make it more likely that the plants would create proteins that people are more allergic to, for instance? I'm not a conspiracy theorist or even anti-GMO. I think it has its place. I'm amazed by some of the stuff that's going on in medicine. It's wonderful. But I do remember a decade or so ago when there was a recall on several foods that turned out to contain GE feed corn that people were having fairly severe allergic reactions to, star something or other. I just have to wonder about anything that we're going to eat, and I haven't seen any really in-depth discussion of how these things work other than to hear proponents say, oh, all the DNA is digested, it can't hurt you. That's not going to fix any problems we might have dealing with new proteins, is it? And that's without even getting into the containment problems, which is another issue entirely. I won't bother you with that discussion.
So I can take a crack at
Sure.
this. Is my sound quality good? You're siloning a little bit, Vince, but
No, you sound great.
You sound good to me.
okay, cool. So the first of all, the older techniques that some agricultural companies were using that are more random, that's also known as the entire history of human agriculture. So the way of getting new genes into seeds and growing them up for the past 10,000 years has been to cross plants until you get the traits that you want, and we didn't know about genes until very recently. And genetic modification that we do now, whether it's in plants or in animal cells, is a much more precise movement of stuff from one species to another. So in terms of which technology is being used, there are differences in the approach, but I don't know that it's that I would call it primitive, what, say, a company like Monsanto is doing. And the
techniques that are being used are, yes, basically the same techniques as people use in medicine. It's all just recombinant DNA technology. It's no big deal.
Yeah, exactly. So you have to do different things to get things into plant cells, but it's all the same.
Fundamentally the same thing.
Yeah, it's gene therapy for plants.
Right.
And then in terms of the of concerns about genetically modified foods, this was the first major public relations disaster for the biotech industry, was the way they handled this. So there's a lot of misinformation and questionable information out there, and some of it seems to have seeped into this letter. This Starlink corn was actually a huge PR disaster, but it did not, in fact, hurt anybody. There's no evidence that anybody was harmed. So when you say people were having fairly severe allergic reactions, no. There's there were about 24 people who came forward claiming to have had allergic reactions. Nobody was able to determine with any degree of specificity that was due to Starlink corn, and none of those were severe. But it did highlight the fact that you can't separate feed corn from food corn within our current food supply, and that it was probably a bad idea for the USDA to have approved that product, because it was a product that was supposed to only go into feed corn, because theoretically it could cause some allergic reactions in some people, but it ended up going into food, including taco shells, I think. And so ultimately they pulled it all off the market. It hasn't been detected in anything since, and it doesn't seem to have done any harm to anything except biotech's reputation. But in terms of real concerns, I mean, I think a lot of that could have been handled a lot more transparently and with a lot more public discussion. But I'm not at all convinced, and in fact, I haven't seen any credible evidence that there's actually any harm to this stuff. So yeah, maybe it was introduced without the level of public discussion that should have taken place. But on the other hand, in retrospect, it seems to have worked out okay. And the other thing to bear in mind is we're not looking at a world where you have a choice between these genetically modified pest-resistant plants and some beautiful, organically grown, no pesticide, no evil chemicals, no nothing type of food. In fact, with more than 6 billion people on the planet, what we have is a choice between these pesticide-resistant genetically modified plants or plants that are grown with petrochemical pesticides. And we know for a fact that those pesticides are incredibly toxic. So I consider the genetically modified foods actually kind of a step in the right direction, because at least they're not blatantly toxic. Maybe that's not perfect, but it seems like kind of a move in the right direction.
All right.
Good. All right, she goes on. I do have a comment on vaccination, though. I do happen to be one of those people who've taken advantage of the philosophical exemption for my kids, but not for the big childhood diseases. Don't worry. They were caught up on everything required when they started preschool except for Hep B. The Hep B vaccine is routinely given to newborns even if their mother tests negative. Even our pediatrician suggested we wait. I don't feel like I'm putting my kids or anyone else's kids at very much risk by not vaccinating them for a sexually transmitted disease during their infancy, and I've been told there are risks associated with vaccinating newborns with undiagnosed heart problems. I just wanted to let you guys know that there are other reasons besides fears of autism for skipping some vaccines. Maybe my judgment of probabilities was off. If so, I guess I'm just lucky. Things have worked out, but that's the way it goes a lot of the time. A lot of us are out here doing our best, going over things as rationally as we can, and still make mistakes. The CDC is judging probabilities that something will happen to a huge group of people. We sometimes then have to figure out where we're going to fit individually among the statistics. Maybe it's not mathematically correct to decide my kids are unlikely to be exposed to something in their first few years of life, but there you go. Love the show. You guys teach me a lot. Suzanne, I just want to know, Suzanne, if you looked at the papers that said there were problems vaccinating newborns with undiagnosed heart problems, or did you just hear it? Because that's one of the problems that people have, is they hear something, but they don't actually go look at it.
And they don't actually check to see if there are papers on it.
Right. So there might not be.
So the hepatitis vaccine, the reason, first of all, as you point out here, Vincent, HPV is transmitted by many other ways than just sex.
Mm-hmm.
Injection, drug use, birth to an infected mother, contact with blood or open sores of an infected person, needle sticks or sharp instrument exposures, sharing items such as razors or toothbrushes with an infected person. And one of the reasons, so Hep B is similar in some ways to Hep C, in that you can get an acute infection and that'll resolve and you're okay sometimes, or if you're unlucky, you can get a chronic infection. And a chronic infection, you can either have repeated episodes of disease or none for a long time, and quite often in the long run, the ultimate result is hepatocellular carcinoma. Not only that, but in the meantime, of course, as a carrier, and maybe even an asymptomatic carrier, you're spreading it around all the time. And the incidence, the risk of establishing a chronic infection is very significantly higher if you are infected as a newborn, relative to being infected later in life. And I believe, I'm not sure of this, I haven't looked at the primary stuff recently, but I believe that that's the motivation for vaccinating people as newborns. Obviously, there's it's obviously indicated if the mother has hepatitis B, but in general, if you are exposed to Hep B as a newborn or at a very young age, your risk of a really serious disease is much higher than if you're exposed later on. And you can get it by mechanisms other than sex. So I think that's where that recommendation comes from. Plus, this is a vaccine where all you're doing is injecting a protein. There's no virus. There's I haven't read the label, so I don't know what the adverse consequences might be, but my guess is that there are very few and very low. So it's as vaccines go, this is one of the safest around.
I'm surprised your pediatrician suggested to wait.
I'm very surprised at that.
Yeah, I'm surprised too.
Because all three of our kids were immunized very close after birth, and pediatrician said it's really important because you can be infected.
I do want to, there's something that I've kind of wanted to discuss, because we talk about frequently this idea of people making personal decisions about vaccines against the recommendations of their doctors and the public health officials and stuff like that. I can understand that it's tough, because you see a lot of stuff in the news media about various medical treatments and whether or not they're effective and whether or not you should do them. I think one of the classics in my mind is hormone replacement therapy. There's a lot of controversy about whether that's a good thing to do or, in fact, a bad thing to do. And people are left in the position lots of times of making actually personal decisions about their health care. And people are told, to take personal responsibility for your health. So I can understand a lot of people being in kind of a quandary about this. Am I supposed to just, about vaccines, am I supposed to just believe everything you say? And about some of these other things, I'm supposed to try and understand what's out there, at least in the press? It's a tough situation.
But Rich, isn't the point though that you have the CDC, you have the American, what is the vaccine
Advisory Committee on Immunization Practices, ACIP.
Right.
Yeah.
So you have physicians and scientists, vaccine people looking at all the info and making a decision. Not everyone can look at all those data and make their own decision. That's the point of having someone make a decision for you, right?
So
and I think a lot of people don't understand that the quality of the science on these things is radically different. So there is a lot that's unsettled about hormone therapy and, for that matter, hormone biology with aging. And the question of, is hormone therapy appropriate? It's a very complex biological issue, and the literature on it is not at all definitive. The question about vaccines is a completely different universe. The literature on it is completely definitive, and there's not really a lot of ambiguity about any of it.
Yeah.
Great. So I'm glad we're having this discussion, because I don't think a lot of people make that distinction. Okay.
The other thing is, infectious disease is not a personal issue.
Right.
It is, vaccines are a public good. And so when you're making these decisions, you have to not just think about yourself, which I know our culture values thinking about yourself 24/7, but you have to imagine, what if I universalize my actions? What if everybody did it this way? Because the presumption is that if you're going to do things this way, everybody should be able to. So if you choose to delay or skip vaccines for your kids, what if everybody did that?
And we're going to see some of that happening.
Actually, the experiment's been done, I'm sad to report, and Minnesota is now dealing with the consequences, and some other places too.
I'm waiting for Ashland, Oregon. Apparently, their vaccination rate is only about 70%, because there's a very large cadre of anti-vaccination people there, and that's a disaster waiting to happen.
Indeed.
Yeah, I think the problem is being put at risk by people who decide not to immunize. I don't think that's fair to everyone.
Yeah, it's like people who decide not to have the brakes in their car serviced.
I remember when we were still using oral polio vaccine in this country. My 3 children all received it, and I remember being in the office when the physician said, no, it was a choice at the time, you can have OPV or the inactivated. She said, you sure you want OPV, right? I said, yep. And I knew the risks, and I was willing to take it because I felt it was a better vaccine for everybody. And I said, yeah, my kids could be paralyzed, but I have to think this way because it's better.
Yep. So, GMO foods, okay. Get vaccinated.
Yes.
Yeah, get vaccinated.
Thank you,
Although,
Suzanne.
guys, if the pediatrician says you could delay it, what do? I don't blame her.
Yeah, exactly.
She's not going to argue against the pediatrician. And I
don't know how the conversation went. I don't know who brought it up.
Yeah, okay.
But it is disturbing that there's a pediatrician out there saying that.
Oh, there are probably many.
Yeah.
All right, we're going to get some flack, I'm sure. But
fine.
That's fine. The next one is from Erin. I'm sure you guys are receiving a constant slew of complimentative emails on a daily basis. I would like to add to them. I recently finished my first degree at Belmont University here in Nashville, Tennessee. During my time here, I found a passion for science through basic general education courses. Immediately, I decided I should take science courses as all my electives in order to see if I enjoyed the tougher ones, but was not able to fit many in. As a student of the humanities, I had little idea of where to start scientifically or what options are available. Since my interests became clear, I began asking everyone about their experiences. I don't say all that to tell my story, but rather to let you guys know that you have been one of these very valuable resources. I have always had a fascination with your areas of expertise, but never really thought about them as a whole. Furthermore, I have struggled with finding a way to really benefit and help other people benefit from both my love of language, culture, and science. It actually caused me to feel quite depressed. I felt limited to either medicine or a more global path without scientific involvement. Then comes TWiV. Your program and topics are the perfect example of how important every area of education, research, and medicine is. The discussion motivates me to continue on this path and gives me assurance that my ultimate goal of helping promote global awareness of such topics is doable and worth working for. I hope I can use my experiences from traveling in language studies, German, Spanish, and Portuguese, to help promote this sort of knowledge globally and also to help be an ambassador between the always adjoined clinical and research-related fields. As for the content of your program, it is the perfect balance of facts, personal experiences, and opinions. If a German student with little scientific background can follow and comprehend such topics through you guys, anyone can. I cannot get enough of the podcast. I have actually had to set an alarm while listening in order to remind myself that I need to stop and study. Thanks for taking the time to read this. I am overjoyed to get insight into a completely foreign world as far as science and academics go. Well, that's wonderful. Thank you, Aaron.
Yeah,
Thank
great.
you.
Thanks very much.
Next one is from Mitchell. Hi, TWiV people. I've heard many discussions on your show regarding the cold chain for vaccines. I found this article today about a company that has a fridge-free flu vaccine that is currently stable for 1 year. There are promising preclinical results, and they are hoping to enter real clinical trials in a year or so. Preliminary but exciting. He sent a link, but it's broken. But we did talk about this on TWiV 70. I don't know if you guys remember, but putting vaccines in these sugar-coated filters, right? And they sort of,
right.
The sugar preserves them.
Mm-hmm.
And I think that is very close to be, at the time, 70 is a long time ago. That was 2010 February. So it could be what you're talking about. But yes, I think this is really moving forward. And we should have used the cold chain picture for this episode, I guess. All right, the next one is from Alex. I was sitting in on a lecture on the modes of infection. The lecture began with many relationships between humans and organisms described below. Symbiosis benefits only the human, no harm to the organism. Mutualism benefits the human and the organism. Commensalism benefits only the organism, no harm to human. Pathogenicity benefits the organism, harms the human. My mind briefly escaped the lecture to wonder whether whole structure viruses ever actually benefit the human in a symbiotic or mutualistic fashion. I've heard of opportunistic bacteria which compete for nutrients and space on the human body, preventing the establishment of more pathogenic species. After class, I went to explore TWiV to find the latest podcast, Viruses That Make You Better, number 124. The discussion of Tanapox virus was remarkable, but you suggested that the virus only creates the TNF antagonist to evade the immune response. To your knowledge, is that any virus which provide long-term purposeful benefit to the host? I know you have explained thoroughly that the best virus is not necessarily the most fatal. You want to stop there and take that?
Yeah, I can think of one example that we've discussed before, and that is it's in a kind of a model system, a mouse gamma herpes virus that the infection renders the mouse more resistant to some bacterial infections. I think I'm remembering that correctly.
Yeah, is it Listeria? Is that?
Yes, I think that's right. So the idea there, and it's actually related to this Tanapox story, because the Well, no, not really, because I think the mechanism is supposed to be that this latent infection by the herpes virus basically stimulates the immune response in a fashion that boosts the mouse's immunity against other pathogens.
Yeah.
So there's at least one example of that, and my guess is many more.
Listeria monocytogenes, yep. Yeah, there are probably more. In plants, there are tons of examples of that. If you go back to Marilyn Rusink in our TWiV at Bozeman last year, she was talking about that. And she has published a review recently, we'll link to it, where she talks about all kinds of symbioses in plants. So I don't think there's anything unique about plant-virus relationships. I think it's probably in us as well. Finally, I would like to sincerely thank you of the foundation of TWiV. I am a student currently finishing up my undergraduate degree in biomedical sciences at Grand Valley University in Western Michigan. Next year, I will be starting a master's degree with an emphasis in microbiology in London. I am finding the content, particularly Virology 101, extremely helpful for preparation. I know, as evidenced by your mailbag, that hundreds of others have discovered the same. Again, this is a remarkable service to the public. You guys are truly inspiring the next generation of microbiologists. Thanks, Alex.
Cool. And thank you again. I hope that's all true. That's good.
Thank you.
Makes it worthwhile. Not that it's not worthwhile anyway, but makes it extra worthwhile.
All right, let's do one more. Hello all. I was just listening to TWiV 125, in which one of the discussion points was the co-option of cafeteria infection centers by the smaller Ma virus. I was struck by the commonalities between Ma virus or May virus and defective interfering particles, since DI particles too can interfere with production of particles with full-length genomes. This has prompted a few questions or thoughts that I would love to hear your thoughts on. To start, if a DI is like a parasite of a virus, could they be a viable treatment for an infection? Alone, DI particles are not infectious, and they can specifically interfere with production of their progenitor virus if they get into the same cell. Do you know of any studies where this has been tried? This parallel has also made me start rethinking the life cycle of a DI. I had always thought of DIs as being gained and lost continuously during infection, but I wonder if there are particularly successful DIs that are continuously maintained more like an independent parasitic virus. Would it even maybe be possible for a DI particle to change the virus that it's dependent on? I.e., could it jump to infect another similar virus? You could then end up having distinct genetic content between a DI and the viral population it infects. I recently went to a lecture by Marco Vignuzzi of the Pasteur Institute, where he said in passing that DIs could theoretically recombine with full-length viruses, acting as a source of new mutations. If DIs could jump from virus to virus, and there were recombination, then just as how viral infections has led to lateral transfer of genetic information, as in bacteria, DIs or viruses of viruses might have similarly helped pass along viral genes. Thanks so much for the inspiration. This is exactly why I'm a regular listener of the podcast.
That was from Anna.
MSc candidate, University of Ottawa. Well, to talk about DIs and disease, so many, many years ago in the 60s, this was really a hot topic, whether you could use DI particles to change the process of an infection. I don't know if you remember that era, Rich.
I do not. Okay. This is news to me.
A big proponent of that was Alice Wong, who thought you could use DIs, and it didn't really ever work, and that was really the end. They fell really hard, and no one has really looked at it again. Okay, so I think the idea that you could use them therapeutically has been visited, and it doesn't work.
Well, I guess a major limitation of it would be the DI particles can only infect infected cells, so you're naturally going to have a point at which they're successful enough, you get that classic predator-prey oscillation,
yeah.
And then, the DI particle goes extinct.
Right. Okay, how about the other part, gene transfer?
I mean, my first reaction was nah, but then I'm thinking, well, she's got a point here.
Yeah.
It's an interesting idea. I don't know of any examples, but it's not out of the question, it seems to me, that you could have a DI particle that could infect is successfully replicated in a cell, replicating a related virus. I mean, DIs are basically origins of replication, right? little replicons. Okay, so as long as they could be acted on by a related virus, they could carry along stuff and maybe recombine, cause gene transfer. I don't see why not. But I don't know. Do you know of any experiments in this regard, Vincent?
No, I don't know of any. The problem is DIs are mostly done in cell culture. I don't know of any found naturally in animals.
Right.
So now the virophages show that a similar particle can exist in nature, right? Mm-hmm. I don't know if you would call those DIs because they are
call those DIs.
They can, although they need to have another virus in which to replicate their cell in which to replicate.
Maybe they are DIs.
Yeah, maybe
right?
they are. Yeah, they do interfere.
Maybe they used to be a cafeteria
virus. And they're out there in nature, sure.
Yeah, and I kind of suspect we're going to find more of these now that the deep sequencing and efforts like that are available. Because one of the reasons that we don't have a whole lot of DIs to study is that, it's just an accident that you find them.
Yeah, that's true. So
they happen to occur in a virus that somebody happened to be studying because it was culturable.
Yeah. No, I think that this could be. We don't have evidence, but it could absolutely be that they're out there and they exchange genetic information for sure.
Yeah. Good thinking, Anna.
Yeah.
Yes.
Good.
Well done. Keep it up.
Yeah.
The life cycle of a DI. That's a good name for an episode. All right, let's do some picks. Start with Rich.
Right. This is a pick that my beekeeper virologist friend Megan sent me. And it's an interactive from the New York Times some time ago about artificial intelligence. And the format is that you can play rock, paper, scissors with a computer. All right. So you look at this. On the left, there's a human hand, and on the right, there's a robotic hand. And you get to choose whether you're going to throw rock, paper, or scissors. And the computer responds, and it keeps score. Right. And you can start off as a novice. That is, the computer is a novice. It's never seen any of this. And you can have it reveal to you what the computer is learning as you go through this. And then you can also play against an experienced computer, and the experienced computer does a better job than the novice computer. And you can see why, because it will, if you run the experienced computer mode, it will tell you what the computer is thinking, right? So I threw paper here. Let me see. I got to go again. It says, come on, show me what it's thinking here. Oh, it's not showing me what it's thinking. At any rate,
that's cool.
Very cool. It'll show you how the computer learned to try and beat you. So
nice.
I thought it was fun to play with.
That is cool. Alan, what do you have?
My pick. Oh, sorry,
You're busy
scoring 1 to 3
paper.
to 3 right now on rock paper scissors. My pick is webcitation.org. And I just found out about this, and it actually addresses a problem that I've been wondering about for years. People writing papers or websites, but it especially comes up when you're writing something for publication in a more traditional format, like a journal article, like a news article. You cite a website, and you say, this information is here. Click the link. Well, the trouble is, you go back to these articles even 6 months or a year later, and the links are dead.
Yeah, we just experienced that.
Yeah, we just experienced that. Somebody sent us a letter, and by the time we answered it, the link was a 404. So this is an inherent problem in writing anything these days if you're going to cite something online. And it's a huge problem in the scientific literature because we're amassing all this knowledge, and it's all stored online, and then it all moves around, so you can't find it again. This site is a nonprofit organization that has set up to archive websites. And so what you do is you, anytime you're going to cite some site, you can go here, archive the URL. And it will take a snapshot of the page as it is right at that moment and save it online. And it'll then give you a permanent URL, which, of course, you're assuming that webcitation.org will continue to exist 10 years from now, which is there's a little probabilistic argument there. But But they do seem to be above board, and they seem to have the right idea. So hopefully these links will last more than the usual few months that a typical internet link will. And so you can do this with anything that you're writing or referencing.
That's cool. But how are they going to make money to keep this going?
It's a .org. They take donations, I believe, and they're just basically going to operate as a nonprofit, I think.
Yeah, it's a great idea.
Oh, yeah, good idea.
My pick is an article in Emerging Infectious Diseases. It's called The Edward Jenner Museum. It's sort of a walkthrough of Edward Jenner's home in England.
Nice.
I've never visited. So
I have.
If I have, I would say I'd pick the museum. You have. It must be cool. It looks great in this article.
It's very cool.
And so the article is nice. And if you have been there, well, if you go to England, go to this museum. It looks really cool. It's small and got all his stuff there. And the article
it's got his vaccination hut out back.
That's right. The cottage used for vaccinating children. Any bifurcated needles?
No, they weren't invented to the 60s.
You just used a needle of some kind, I guess?
Yeah, they had special tools that they did this with. I forget what I could probably dig up one, find out what it looks like.
While standing in the church talking with us, the vicar noticed a woman quietly waiting for him to finish. He asked if he could help her, and she said she was looking for the grave of an ancestor. I asked, "You mean the milkmaid?" And she said, "Yes."
Ooh.
Isn't that cool?
Wow.
There you go. We also have a listener pick of the week. This one is from Derek Tolley, who sent it on Twitter. He found a documentary called A Paralyzing Fear, The Story of Polio in America. Then he found it on Netflix, but you could probably find it any place that has video. And in fact, there's a book that accompanies this documentary, came out a number of years ago, full of interviews that were used to make the documentary. So they're both very good. So thanks for that, Derek, our listener pick of the week. And you can send yours in any way you'd like, and we'll put them up here. And that will do it for another TWiV. Don't forget Facebook.com, This Week in Virology. Check out all our other stuff that we do there. You can find us on iTunes at the Zune Marketplace or at twiv.tv. And there's an app for your iPhone or Android device at microbeworld.org/app. If you are new to TWiV, go on over to iTunes and leave a comment. It helps us to stay on the front page of the medicine podcast directory there. As always, send us your questions and comments to TWiV at twiv.tv. Rich Condit, down in Florida, thanks for joining us.
Great to be here. Another good time.
Enjoy your golf.
Oh, I will.
I'm sure.
It's going to be a great event.
Alan Dove is at alandove.com. Thank you, Alan.
Always a pleasure.
And last week we were reading our email, and one of them was from an Ina, right?
Ina.
Ina.
Ina or Ina?
I don't know. But you said you don't see that name very often, and I just realized that's one of your uncles or aunts or whatever.
It was my grandmother.
Your grandmother. That's your blog there that you're posting.
Yeah, the Walter and Ina site.
I realized that after we stopped recording, so that was pretty cool. That's a person with the same name.
Yes.
There you go. I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.